So this is Natural Science Seminar. It's the first one of the year. One of the challenges that we have of doing natural science seminars is getting speakers that are going to be able to connect with such a broad audience. We have, how many biology freshmen do we have in here? Raise your hand proudly. Okay, we got some of you. How many senior seminar students do we have in here? Biology or chemistry? You're signed up for it. All right, good. So that is a very broad range of understanding of chemistry and understanding of science in general. So the trick is to make presentations that will appeal to a broad audience. That's why you see a lot of students over here. They're not just decoration and fluff for the main event, Dr. Kuntzleman, right? Dr. Kuntzleman understands this disparity in ability, so he's going to try to present some material that will be graspable by you, the student, on different levels. So some of you may need to use math, okay? I know that's a dirty four-letter word, but sometimes we say that word around here. Some of you are more like me. You're having difficulty with math, and it's really challenging, and so you want something that's more conceptual. So this one is going to be kind of in between. You can do a lot of things with the types of presentations that Dr. K is great at doing. In fact, Dr. K is so good at it that his ideas are on the cover of an international journal of chemical education. So I've seen this journal on the stacks in Japan. And I know it's on the stacks in Russia, and I know it's on the stacks in China. This is truly a momentous situation to have two journal covers for such an international journal, and they are due to Dr. Kuntzleman's creative brain. So, please welcome Dr. Kuntzleman, and he'll do the other introduction.
Well, thanks and welcome to the first science seminar. And Dr. Baldwin might say this is due to just my creative brain, but I think all of you know, if you've had me in a course, that I get a lot of mileage out of you, the students. And also from picking Dr. Baldwin's brain, Dr. Baradovich's brain, Dr. Newhouse's brain, and I look forward to picking Dr. Wyman's brain as well. So a lot of what you see today are going to be experiments that we have designed here at Spring Arbor University with students, with biology students, with chemistry students, and even with some art students, in order to try to make chemistry come alive a little bit, make it sort of interesting. So the title of the talk is Lightsticks, Camera, Action. The idea behind that title is two different experiments that we've designed, and both have been featured on the cover of the Journal of Chemical Education. The first deals with Lightsticks, and Kristen Rohr has done an enormous amount of work, as well as some other students, and even Dr. Baldwin has helped out with these experiments. This picture here that was on the cover, I don't know if Cassie Lance is here today, but she actually took these pictures that ended up on the cover. And then the second group of experiments deals with a stunt, it's an actual stunt, the chemistry behind a stunt that's done in the special effects industry. Is how do people safely light themselves on fire in the movies? And so that's the camera action portion of the talk. And this is a picture of my hand here with a little bit of fuel.
He's world famous for his
And this picture was taken, I don't know if any of you know Keegan Adriance. He was an art student. He took this picture for us. Can I go so much slide for this thing?
No, you can't.
Give me another one. So we're involved here at Spring Arbor University with a lot of outreach to the community. We try to bring science to children and even their parents in some instances. So these are some pictures of some of the things that we do here at Spring Arbor. This is an advertisement. I'm trying to recruit you to get involved.
Yay!
Okay? So these are some pictures of our summer science camp, these four around here. and that's usually held in late June of the year. And we often get education students involved to actually be teachers. They get classroom experience working at our summer science camp, and we do a lot of experimental, trying to design experiments for camp, and a lot of those turn into some of the experiments that you're going to see today. On this side are some pictures, and here in the middle are of our Halloween and the Science Lab celebration. This year that's going to be Wednesday, October 24th. That starts off with a chemistry demonstration show that lasts about a half an hour. Me talking at the show there. And then once the show's over, the students are released into the science building and they go trick-or-treating. They wear their costumes. They trick-or-treat through the building, but instead of actually getting candy when they go to a room, they get to perform a science experiment. And when they're done, they get a little piece of candy and they can move on to the next place. This is our, well, this will be spelled right. That should be natural. But this is our t-shirt design that we're going to have for science camp in the coming year. And you can also order one of these if you like in the main office. Okay, so we're going to get started. I'm going to start off first. We'll deal with chemistry of Lightsticks, which Kristen's involved. And then we're going to talk about the special effects stunt. And I have Chris Minier Spencer, we got Levi Norris, and we have Dakota Moore, who have been working on extending this project, and hopefully in hopes of getting another publication out. So, we have here a representation of the, there should be an arrow there, of the chemical reaction that goes on inside a light stick. So in order for something to emit light, that something has to have energy. It's got to get energy from somewhere. So when you activate a light stick, what's going on is you are mixing chemicals. Those chemicals go through a reaction. Sure, we can just get it going here. So when I snap the light stick, what I'm doing is I'm breaking an inner glass vial that has some chemicals in it. And then that mixes with some chemicals outside of the vial. And when all those reactants mix, you get a chemical reaction. And that chemical reaction is gained by dyes that are placed inside the light stick. And then those dyes release the energy as light. So these are different representations. You might understand some, you might not understand. They're all different ways of saying the same thing. If you've had organic, you know what these things are. Okay, this is a phenyl oxalate molecule. This reacts with hydrogen peroxide. Same kind of stuff you got in the brown bottle in your bathroom. In the presence of a base, the base is a catalyst, gets the reaction going. It makes it go quicker. And then it produces this molecule called phenol. We're not really interested in that. It also produces some carbon dioxide gas. We'll show you that in a little bit. We're not really interested in this either. This is what we're interested in. We're interested in the energy. Here's another representation of the same thing. Okay, these are just the chemical formulas for all of these molecules here, and this is written in word form here. So here's just a diagram of where the components are found in a light stick. This represents an inner glass vial. Inside the vial are the phenyl oxalate and some dyes and just some solvent to mix it in. Outside the glass vial, you have your peroxide, your base, and your solvent. When you crack it, the things mix, and off it goes. If you don't have the dye in the light stick, the energy is just given off as heat. The light stick will just get hot. But since there's dyes in the light stick, those dyes gain that chemical energy, and then they give that chemical energy off as light. Let's fiddle around with this a little bit. These are just some actual different fluorescent dyes. I've bolded them with the different colors that they've given off. You might not be surprised to hear that different dyes are used in different colored markers, right? That makes sense. Well, it's no different in a light stick. Different dyes give off different colors of light, and these are just different chemical structures. If you've had organic chemistry, you probably recognize these things. You might, God forbid, even be able to name those things. I certainly wouldn't be able to do that. It's not stuff I think about like Dr. Baldwin. Anyway, I bolded each one in the various colors that they emit. So if we take, for example, a let's do a green light stick here. Whoop. And let's get this machine going here. This is kind of fun. What this machine is a fiber optic. And you can see if I'm pointing at the light, you see it go up, and then if I cover the fiber optic, it disappears. So I'm pointing it at this light up here. I'll catch it just right. Oh, there it goes. There we go. Okay? What this is doing, it's measuring the wavelengths or the colors of light that are being given off by this light up here. And because it's basically giving off, on the bottom, if you look from 400 to 800 nanometers, that's basically visible light. That's all the different colors of visible light. The thing looks white. All the colors of light together basically look white. Let's try green here. You can see your emission profile for a green light stick, and you can see that green runs around 520, 530 nanometers of light. And you try red. Red here. Red runs somewhere between 650, 700. You can also see the emission of light kind of tailing off. It's not quite as bright as the reaction goes on. We'll try yellow. Really up there. Yellow usually runs around 580, 575. A little bit of blue. Blue's down below 500, usually in the 450 region. And the reason why these are different colors is they put these different dyes in them. They actually have different dyes in the light stick mixture that when they gain chemical energy, they're just going to give off different colors of light. Whoop. Let's do a quick experiment here with our green light stick. What I'm going to do is I'm going to take this green light stick and I'm just going to demonstrate to you first, I hope, if it works, sometimes it doesn't always work, that a green light stick does, any light stick does actually give off carbon dioxide. Can I use that or have I got to use a different one? That's mine, okay. I'm just going to pour that in there. Okay. And then what I'm going to do is I'm going to add a little bit of the catalyst that is used, we'll get the lights there, Nate. A little bit of the catalyst that is used in the light stick mixture. The catalyst is sodium salicylate. A catalyst speeds up a chemical reaction. It makes it go faster. So we're going to add just a sniff of this stuff. Put it in there, mix it, and sure enough it gets brighter. Let that set a little while. You might notice some effervescence if you're sitting up front. You see how it kind of looks like it's expanding a little bit? Those are from some bubbles that are being produced. The bubbles are from carbon dioxide. That's one of the products that's produced in the chemical reaction. Now you might also notice, as it's done fizzing, it's getting more dim now. That's because you add too much catalyst, the reaction's going to go really fast, and it's going to burn out quickly. So if you're a chemist and you're trying to design a light stick, you better engineer that light stick with just the right concentration of catalyst in the light stick. Otherwise, the kid's going to break the light stick, and it's going to be really bright, and he'll get really excited, and then it'll go out, and he'll be mad. Or she'll be mad, whatever the case may be. So chemists need to think about this, right? You've got to think about what kind of concentration am I going to use so as to make this light stick burn just the right amount of time. Why don't you guys feel the bottom of that for me? Why don't you tell me what that feels like? It's what? It's warm. Okay? I can't go around the whole room doing this. I wish I could. But this reaction, contrary to popular belief, a lot of people think that light sticks just give off light and no energy as heat. Well, I just made this light stick reaction go really quickly. And it released a lot of light, but at the same time, it was able to release enough heat that folks could feel it. And normally that's, in a regular light stick, you can't. The reaction just goes so slowly, it releases heat in such small incremental amounts that you don't really notice it. I've done experiments with regular light sticks to show that sure enough, if you find the temperature of these things, they do gradually go up about a degree Celsius or 2 for every 2 hours. They do rise in temperature. All right. Current slide. Now, some things we've been working on here at Spring Arbor is to, go ahead and hit the light there, Nate, is to try to show the role of different dyes in the light sticks. So just like these different dyes that I showed on the previous slide give off different colors of light, there's an enormous number of dyes, an unbelievably large number of dyes that give off light in this manner. And we have a couple of them up here on the screen. Anyway, the one that's bolded in red, that's chlorophyll. You biologists, I might be speaking your language now. That's a pigment found in plants. You may or may not realize that chlorophyll is one of these molecules, thanks Dr. Baldwin, that can give off light. We usually call that fluorescence. In this particular reaction, when we put it in the light stick reaction, it's called chemiluminescence. But this molecule actually will give off light, and it will give it off in the light stick reaction. You want to do that now, or you want me to talk about the others? Kristen right now is actually preparing a extract of chlorophyll, and all she's doing is taking some spinach, putting it in acetone. And when you do that, you get that beautiful green solution that's indicative of chlorophyll. Chlorophyll fluoresces red. All right, so what she's doing is she's adding the chlorophyll extract, nice beautiful green color, to the test tube. And then what we're going to do, I think we need the lights for this, she's going to add a little bit of the light stick mixture. And sure enough, okay? So there's a dye that you can get in your kitchen, right? You can extract it into nail polish remover, that's what acetone is. And then you can cut a light stick open, pour a little bit of that white light stick mixture in there, and you can get the chlorophyll molecule to give off its red color. And like I said, there's a wide variety of household items that this can be done with. Curcumin is a dye that's found in turmeric powder. Dr. Baldwin uses this a lot, this molecule a lot in the different labs that he uses. You got the curcumin ready? Let's see what color that fluoresces. Curcumin fluoresces a yellow color. Cool. Okay? Beautiful yellow color. Erythrocene. This is a molecule that's found in red food dye. And erythrocene, when it's in solution, it looks red. That's why it's, red food dye, right? No doubt. But if we're going to take that erythrocene and use that in the light stick reaction, let's see what kind of color we get here. So Kristen has added some erythrocene to some acetone. She's going to add some of the white light stick mixture. And we get a beautiful orange color. Food dye. Food dye, everyone's got food dye somewhere. A little bit of nail polish remover, acetone. Got that at your house. You want to cut a light stick open and try it. It's going to work. It's going to work. Very simple reaction to run. Finally, the molecule quinine is found in tonic water. It's also found in these pills that are used for leg cramps. Some folks like to drink quinine because it helps with cramps in your legs. However, recently, I think it was 2010 or 2011, the FDA has shown that that might cause some health problems if folks are taking, not drinking the tonic water, but actually taking the leg cramps pills. So I used to be able to buy leg cramps pills with quinine in them at the pharmacy. I can't find them anymore. I haven't been able to. So if anyone knows of an easy place to find quinine other than tonic water, let me know. We've tried this experiment with tonic water. It hasn't been able to work, but we have got it to work with the leg cramps pills. And I still have a few of them left. You got some there?
Yep.
Got it ready to roll? Almost?
Yep.
So Kristen's taking the leg cramp pill with the quinine, and you get a nice blue-purple excuse me, blue-violet colored fluorescence. All right. So these are just a few of some of the, yeah, we can do the markers. These are some of the other household items, just a few, that we've been able to find that emit different colors in this particular experiment. So we've used spinach here, we showed you erythrocene and different food dyes, curcumin and quinine. We've also found that we can get emission from dry erase markers. They have some dyes in there that fluoresce. There's a wide variety of other sources of household items we found that fluoresce in this particular reaction. We'll just show you the markers for now. Before, I guess while she's getting those ready. You all set? All right, we'll wait here. And I'm always looking for new ideas for sources of dyes that will fluoresce in this particular reaction.
Can you predict chemically what's going to fluoresce, what isn't?
Oh man, what a hard question. You want to feel that, or you want me to feel that?
I think you can probably feel that a little better. Usually, this is my answer, and then I'll defer to the expert. My understanding is usually molecules that tend to have a lot of rings in them and that tend to be flat are molecules that tend to fluoresce. So if we go back and look at some of these structures here, and Baldwin can correct me if I'm wrong here. He's Mr. Fluorescence. You see a lot of these ring-shaped portions of the molecule here. This is a flat series of rings. What we like to say is that the electrons are smeared all over the molecule, and they're able to move back and forth around the molecule very easily.
And scientists who tried to design new fluorescent dyes, also, you have to have that long smear two-dimensional surface, but you also have to have one end of the molecule that's electron dense, and another end of the molecule, the same molecule, that's electron deficient. And when you have that imbalance set up, that tends to be a formula for making a fluorescent compound. What color is very difficult to predict. That's when you go into the laboratory, you've heard of the laboratory, and you do the experiment. Then you find out for sure. I don't like these people that are armchair chemists and just sit around and calculate things on their supercomputer. I'm a lab guy.
All right, I think Kristen's got the markers ready here. So what color is this one?
Red.
This is a red fluorescent marker. With a pink color. We can add some, I'll add some of the base to get it going.
Oh yeah, that really burst.
What's that one?
It's the orange.
Orange. There we go. What color is that marker?
Yellow.
It's a yellow one. It's got a little yellow green. You mind handing me that one? Let's give this a little sniff of base over there.
That's the green marker.
What marker is that one, Kristen?
The one that he has is green.
The one that you're holding is what color?
Blue.
That's blue. It's not quite as satisfying, huh?
Not yet.
Yeah.
It's kind of dim, isn't it?
Yeah, this one's
You can see it. It's there.
There's your red, huh?
Yeah.
You want me to add a little of that? Did I add some to that one?
Yes.
I did. Probably burning out.
Yep.
Probably.
We'll talk about it next.
There we go.
Yeah, that's great.
Beautiful. We got time for one more thing on the light sticks. Not all the colors come from just one dye. In some cases, you need a mixture of dyes in order to get the color. You might know that when you mix all the colors of light together, you get white light. So we got a white, here we go, white light stick here. Let's just look at the emission profile In other words, what light is being given off by a white light stick. And as you might guess, there's quite a few different dyes. You can see, I see like 1, 2, 3, 4, maybe 5 different peaks, which tells me there's probably about 5 different dyes being used in a white light stick. You throw in a bunch of different colors of light, you get white. That kind of makes sense.
I think I got the other ones.
One of my favorite colors of light sticks is a color changing light stick. You take this one, it starts off a magenta color. You might see it as pink, but it's easy to see that there are 2 different dyes in this particular light stick. One of them emits a red color, the other emits a blue, and to your eyeballs it looks magenta or pink. And what happens over time, if you watch that red peak, which is in between 600 and 700, you see how fast it's decreasing? See how fast that peak is going down? What that's telling me is that that dye is actually being destroyed in the chemical reaction as it emits light. So as it gives off light, it's destroyed, and as that red emitting dye is destroyed, the blue color of dye shines through. So to give you an idea of how this works, there's white. Orange is a mixture of 2 dyes. I'm not going to show that, but these are some experiments that were done by Megan Connors here. She actually was able to separate the 2 colors of dyes from a light stick on a piece of chalk. Purple, you get the red and the blue dye. Aqua, this is another one of Megan's experiments. She was able to separate 2 colors from an aqua light stick. Purple to blue, the way this works is, over time, the red dye fades, and you're left with just seeing the blue. Let's show how that actually works. We can speed this up so you can see it with a catalyst. Got a test tube ready for me? Maybe? So we'll speed this process up with a catalyst so you can actually see it happening very quickly. That's red. Let's see what happens. Got a little dirty test tube. We'll see what happens here. All right, so that's glowing nice. Purple color. Got a little bit of catalyst here. Got it? All right. Let's see what happens. There's my salicylate catalyst. Mix that around. It's really bright. See some effervescence near the top, the bubbles.
Seeing any change?
Might be shifting a little bit up near the top.
Are we just imagining this?
Just watch it. Well, that's really bubbling, huh? Shift to blue.
I think it's starting to shift now.
It's also getting a little dimmer. Now that's not as red as it was before, is it?
No, no. Much less red. Up!
Boy, that's a pesky red in there, isn't it?
Do you
Maybe we should have just done one, huh?
Do you have a picture of that shift?
A picture of it? Anyway, you saw it subtly there. You saw it. All right. All right, let's move on to the next part. Lights on. So another experiment that we've been working on deals with something called fire gel. That's our name for it, fire gel. And sort of the pioneer in this work was Gary Zeller. This is actually Dr. Zeller, using what he called Zell gel. And Zell gel was, he doesn't tell me, I talked to the guy on the phone, he wouldn't tell me what's in it. I called him up and asked him, you know, how do you make this stuff? Because what you do with the Zell gel is you can coat your hand or whatever you want to light on fire with this gel. You spray it with a fuel, and then you light the fuel on fire. And the energy from the burning fuel doesn't go into your hand or your head or wherever you're on fire. it goes into the gel. The gel acts as an insulator. It's simple. It's a very simple idea. And he made this as he extracted material from plants and made this gel that he won't tell anyone what it is, and he won't sell it to you unless he trains you in how to use it. And it costs about $500 a gallon. All right? So he was a pioneer in this and actually won an Oscar for scientific achievement for developing this stuff, because it used to be when folks would light themselves on fire as stunts, they had to use all this bulky clothes and equipment to protect themselves from the flame. And he just has this layer of gel, which is very easy to make it look like there's really nothing on your skin or your hand. Also, there are some folks, if you're interested, you ought to look for these guys on YouTube. They have some interesting experiments that they show online. They also have some interesting language that doesn't really follow the concept, so beware of that. There's some pictures of that on the web as well, but they have been able to develop a gel that does essentially the same thing. And they have been nominated for an Academy Award for their work. And I called these guys and asked them what was in their gel. And of course, it's a well-kept trade secret. They won't let your stuff go for obvious reasons. You know, they don't want idiots like me trying to light themselves on fire. They get in trouble for that. So I think this guy, Dustin Brooks, had the world record for full body burn. It was well over 2 minutes using this particular gel. Well, I was interested in this. I wanted to try to you know, show the chemistry behind how this works. That's what I'm interested in. How is this working? So I got on their website. I'm looking at some of their pictures, and if you look really closely at these pictures here, you notice there's a gel on here, right? You can see their hands are coated with something that looks kind of a little bit like a jelly. And one way that I learn a lot about the way things are done chemically is I search patents. So I started to get into the patent literature, trying to find out what kinds of things were being used to use as a fire protection barrier. And finally I hit it when I searched through the patents, fire gel. And this particular one was very helpful to me. This particular patent described using a super absorbent polymer. I know what that is. I know what that is. I know how to get it. It's used for insulating a person or object from increased temperature. And they talked about various commercial products. Well, I looked all these up, and the only one I could find that would be sold was this Cold Shield Thermal Shield Gel. This stuff here, that costs about $20 a quart. I bought some of this. I remember, I think it was Dr. Newhouse. I first showed him. Went down to his office after many experiments. First, not using my hand, okay? Using thermocouples and shielding the thermocouple with the gel and lighting that on fire and making sure I didn't see much temperature rises. Well, once I was pretty certain that the thing worked, I tried it myself. I showed Dr. Newhouse. And I remember I was standing right down there in his office, and I was so excited about this. And so we started buying this stuff, and I looked at its consistency, and I thought, you know what? We can make that stuff here. So I looked up the Material Safety Data Sheet, MSDS, for Cold Shield. And sure enough, it popped up. And an MSDS is going to tell you, not perfectly, but it'll give you clues as to what's inside the material. So when I did this, we found the major components of Cold Shield are water, clay mineral, commonly used in cosmetic preparations, talc, found in baby powder, xanthan gum, used in food, acrylic polymer, that's the superabsorbent polymer, used in cosmetics and drugs, and you stick a preservative in there, which I don't. My stuff molds over after a while. If you put the preservative in there, it won't. So, first I want Levi here to demonstrate to you what a superabsorbent polymer is. So what we have here in these test tubes are 50 milligrams of the superabsorbent polymer. It's the same polymer that's found in a product that everyone in here, probably 20 and under, has worn at some time in their life. Okay?
Some of us never experienced.
It's found in diapers. So what he's done is he's poured pour it all in there?
No, you want me
Go
to
ahead, pour it all in there. Let's see. He's poured 30 grams, almost, what is that? That's almost 700 times the mass of water into the test tube. Shake that around a little bit. Let's see if that works. Well, maybe not. Maybe I told you to. Oh, there we go. So we had 50 milligrams. We added almost 50 grams of water. That's 1,000 times the mass of water. Let's see what we got there. It absorbs it. It's a super absorbent polymer. Why don't you try that with tap water? So now, the water he just used is deionized water. That's water that has all the ions taken out of it. We're going to try it here with tap water. Try the same thing. We'll let that go for a little bit. It doesn't work as well with the tap water. You biologists know why. Okay? These polymers work on the principle of osmosis. Principle of osmosis. If you remember osmosis, if you have two cells of water separated by a barrier through which water can move, but not ions, okay, the water can move, what's going to happen is the water is going to move so as to make the ratio of water and ions the same on both sides of the container. So if we start, go ahead and back that up, Chris. And if we start here, this side of the container doesn't have many ions. This has a boatload. This has way too much water in it, so the water is naturally going to flow across this barrier into the other container. Oh, back that up a little bit. Until the ratios are the same on both sides. That's osmosis. Really simple. Go ahead and go to the next slide. This is our superabsorbent polymer. So polymers is a really long chain molecule, but what these superabsorbent polymers have on them are ions. They line it up. And the polymer forms a barrier around all these ions. So if you pour water on the outside of a polymer, those things are naturally going to want to go inside, so as to equalize the concentration of ions inside and outside the membrane. If you throw a little salt, why don't you go put a piece of rock salt in the one with the DI water. Let's go ahead and throw some salt on the outside of this animation here. Well, now, outside of our polymer, there's a whole bunch of ions. The water's going to have to move out in order to equalize the concentrations on both sides. Levi has added a single crystal of salt to the test tube that had the water absorbed by the polymer, and you can see it's, you should turn that thing upside down. The water has now escaped the polymer and come out the other way. So that's the properties of this gel that we're trying to use in this fire gel. Let's go ahead and have Dakota here. He's going to sort of show you how this fire gel is made, and we're going to use it. We're going to actually try to use it. So all we do is you take a little bit of, is that deionized or tap?
Yeah.
It matters. We learned that, of course, right? We learned that it matters what type of water you use. And take a little bit of that super absorbent polymer. And this was the trick. We tried all sorts of stuff, and finally, one day it hit me. We'll just blend it. We'll just blend this stuff. You know, we would try all sorts of stuff, and we got all this chunky mess that wouldn't really stick real well to your hand. And one day it finally hit me. Well, it's fit smooth. Let's just, let's just blend it in there. So you got the go for it. So we've added the super absorbent polymer, making a nice, interesting looking gel.
Little by little.
Little by little.
So is this microwave?
This is the only thing I use this particular blender for. Got nothing there. That's quite a bit. Is that going to work?
This one thins it out.
That one thins it out. Okay.
Oh, it works.
Got it.
Okay. I've done this before.
Yeah.
I'll leave it be.
I'll leave it. It's called practice.
These guys are the experts on the lab end. I don't know what's going on. What do you have in there? Is that the baby powder?
Mm-hmm.
Okay.
Well, no, this is baby powder. That's just the rest of it.
Okay.
But we'll be adding the baby powder.
He's moving it to the middle so it will go down.
Yeah. Suppose we should have unplugged it, eh? Now it's been recorded. I don't.
It's got a safety.
Oh, it does. Oh, okay.
Yeah, you gotta have the lid on.
Well,
And right now it's too chunky, so then we add the baby powder.
I didn't know they did it like that. You learn something more every day. No wonder I was having so much trouble making this stuff on my own. Look at that.
Now it's moving.
Not bad. Well, we'll make this like a cooking show. When you're done,
yeah, right.
you get it. All right. I'll give you another couple seconds here. There we go. Good? See how it looks. Not too bad. Not too bad. So you get your gel that sticks to your body pretty well. So we're going to go ahead and show you how this works. And this is very similar if you actually look at some of the experiments on YouTube that Fire for Hire does. You'll see it's done very similarly to what I'm going to do here. In this bucket we have a large amount of fire gel made up. And I'm just going to put my hand in here. Put it in there. Roll it around. It's going to make a little bit of a mess. Pull it out, and you got this nice goopy gel on your hand. I make sure I do everything myself. Let me do that. I'm lighting myself on fire here. I want to make sure that... It is sloppy. I'll make sure I have it coated. Trust me.
You want me to tuck your tie in? Yeah, don't strangle the demonstrator.
All right, I'll blow this out if it starts to hurt. Anything. There we go. Now, what those guys do is very impressive, all right? These guys will actually light their whole hand on fire and wave it around. I'm not that crazy, okay?
Yeah.
I'll go ahead and let this burn out until I feel it, but you know, I'm not a stunt person. I'm a chemist, so might be able to get that burn quite a while. But you'll notice I do this a little longer than those folks do. Because they get quite a flame on their hand if you want to Google that. Let's try that one more time. Once more. Okay, get a little bit more on there.
Is the polymer absorbing the heat, or is it forming a shield, or what's it doing?
The polymer absorbs the water. So the polymer absorbs it. There's an enormous amount of water in this bucket. Okay, but it's been caught inside the polymer like that animation showed.
And so the fire is burning off the water, and that's where the heat is coming?
I don't think the water is boiling as far as I can tell. I just think the energy is going into the water. Now most of it goes straight up.
Right.
Most of the energy goes straight up. Some of it is going to go down into not my hand, it's going to go into the water.
So the
I think
water-
is the best
water is
insulator.
absorbing the heat.
Yep.
It's almost like if you had a
It's an
ice
insulation.
cube on your hand.
That's right. It's just it's like I had a huge cup of water that the fire is burning on.
That's a good one.
I felt it too. I don't want to mess around. And you feel it get a little warm. Okay, I don't play around with that. I mean, it's not like the warmth that I feel is not gonna burn me, but I'm not gonna test that. Okay, I'm not a stunt person. Anyway,
Your wife works in the emergency room, yes?
What's that?
Your wife works in the
My wife works in the emergency room. You might notice I take my wedding ring off for that demonstration because, you know, metal's a good conductor.
Yeah.
So, you know, it's good to have a little bit of scientific knowledge. Oop. At this point, I'd like to get my hand cleaned off and acknowledge. Oh, you mind hitting mute there? Nate, thanks. A lot of the folks that have worked on the Lightstick project. Dr. Baldwin and I have done quite a bit of work on the chromatography of Lightsticks. Stephanie Briseño took some pictures. She's an art student here who did some work a few years ago. Anna Comfort, Megan Connors, I already mentioned Cassie, who took the picture that ended up on the cover. And then our fire gel project, Kyle Groen and Colin Howder, they were students here a while ago. And then Chris Dakota and Levi. I'll take questions at this time, if anybody has any.